those talks seems to be that once we enter life as we know it, any explanatory framework that we have heard right now centers on the phenomenon of regulation in one form or another, whether it's the regulation of development, the ecological regulation, disease as a regulatory mechanism, or our understanding to interfere in the regulatory system if we want to design them. So as a challenge here, and to answer our physicist colleagues here, can regulation become a unifying theoretical framework for biology? Doug?
Yes.
No.
I don't know. Oh, come on, you guys.
It's an interesting question, but it depends a lot on what you mean by regulation and the controls on it, because the degree of regulation that Kevin showed with microRNAs is of a far different quality than any kind of ecological regulation. So it's a pretty loose framework, I think, to try and encompass a whole variety of activities.
On the other hand, there are some aspects of regulation. Homeostasis is an idea we're all familiar with as a foundation for biology. One of the principles of Darwinian medicine that I found most useful is what's called the smoke detector principle. Essentially, we've taken signal detection theory and applied it to the expectation of how natural selection should shape the regulation of defenses, such as fever, pain, nausea, and all the rest. So the question is, you smash your finger with a hammer, and it hurts way more than should be useful, right? And you can know that this is the case because you can take a medication to stop your pain and you don't die because of blocking this natural reaction. The explanation for this is the smoke detector principle, because the cost of that defensive reaction is quite small. The cost of not having a defense reaction, such as a panic attack or diarrhea or whatever, could be death. And as a result, a vast majority of the suffering that our patients experience is perfectly normal, but not necessary in the individual instance, which opens up a whole arena for making life better.
George, do you want?
Nothing to add there.
Okay, so we have not quite agreement on whether regulation becomes the framework of a biological theory, and with that, I would open the floor for questions. Yes?
Too disciplined.
Okay, here. So if for sake of argument, we say that microbial life in the universe is an inevitability on an Earth-like planet with under certain conditions, under conditions that we can accept would lead to origin of life. Is it, in your opinions, inevitable that we should have animal and plant life evolve necessarily out of that initial unicellular life? And the reason I ask this is I'd like to make a linkage to some of our earlier panels discussing origin of life and origin of Earth-like planets and the search for Earth-like planets.
Well, I think it's all about time, isn't it? It's how much time do you have? And that really is this ticking time bomb aspect of, if you have a planet that, we were talking about Earth-like planets this morning. I had to laugh. We were talking about which Earth. I mean, the Earth that we have now is so much different than that Archean Earth, as you know. And so the search for Earth-like planets, is it the complex life Earth-like planet or the microbial Earth-like planet, or which one? But there's so many systems on this planet that keep temperature regulation, as you well know, is a very simple one. And without it, we don't have time to produce complexity. And so it's really the planetary systems, I think, that answer that question. What do you have inherently in the planet as it evolves that's going to give you time or not? And it's that or not, I think, that's been more common than the allowance.
I think I'd like to highlight just the dramatic impact of contingency that it has on this argument. Our work has shown that animals like us, animals with guts and nervous systems, evolved from sponges. That the last common ancestor of all living animals today was designed very much like a modern demosponge without its glass skeleton. So if you went to an invertebrate zoology lab, or just went home and looked at a real bath sponge, if it was actually a Hippospongia, that is actually a very good picture of the last common ancestor of animals. What's interesting is that sponges didn't arise, according to our molecular estimates, until about 750 million years ago. And they arose during a key time in Earth history, during the snowball Earth interval. And we can tell stories, and they're just simply stories at this point, that the impact of freezing over the Earth would have had on the life cycle of a sponge, one of the dramatic results would have been is changing the life cycle of sponges. Because, of course, they have to find favorable habitat in order to settle. And it looks like animals with guts and nervous systems actually evolved out of a sponge larval phase. Now, that was the only time in Earth history where you had this snowball Earth interval. And so if you didn't have that, we wouldn't be here. If you don't have animals, I would contend that you don't have plants because you have nothing driving macroevolutionary arms races, because I think it's primarily driven through macrophagy, at least during at this point in time in Earth history. So if you went to a planet that had sponges, you need not necessarily evolve an intelligent animal that actually preyed on other animals. If sponges lived done just fine, feeding on basically dissolved organic carbon.
Okay, Richard.
I just want to enter a protest against this recurrence of the suggestion that there's something odd going on in the Cambrian and very early times, and that we only got new phyla and classes, then nowadays we get new orders, etc. That's obviously got to be true. I've compared it before to a gardener going into a garden, looking at an old oak tree and saying, isn't it strange? Nowadays we only see little twigs sprouting from this tree. We don't see any great big boughs anymore.
It's not true with plants.
What do you mean?
Most higher plant groups are paraphyletic. Most phyla turned out to be monophyletic. That's the great insight that molecular systematics gave zoology, is that what classical zoologists recognize as a unit of morphology turned out to be real. That's not true in plants.
No, that's an entirely different point. I mean,
No, it's not an entirely different point at all, I don't think.
Plants are the perfect counterexample to the argument. If you look at the evolution of major, I mean, the term body plan is probably a bad one, but the major architectures of plants, they appear sequentially through evolution all the way up to angiosperms. That's
Well, even
not,
dicots.
And even dicots. That's not the pattern that you see in animals. Of course, the other way you can get around this is that we can now completely put aside the issue of phyla and orders and classes and look at the same question quantitatively and measure morphological disparity. And what we see overwhelmingly at the base of the Paleozoic is very early maximal disparity followed by filling in of this morphological space. And that's just an empirical result from quantitative morphometrics.
nevertheless, the null hypothesis ought to be that the major taxonomic units will branch off in the earliest eras. So that should not be surprising. That should be what you'd expect.
No, what is surprising, I think, is the extent of the morphological divergence and the fact that that's clearly not a consequence of extinction. We know that now. It's the pattern of evolution in the Cambrian is one of very rapid and extensive architectural innovation.
I suspect that that's seen with hindsight, that you're classifying them as modern phyla.
I also think that neo-Darwinian theory predicted that disparity is created by the extinction of intermediates. So that means that disparity should increase through geologic time. That's patently not the case. That's what Doug was saying, is that disparity is established very early in a clade's history, and then you fill that with diversity. That's just patently the opposite of what the prediction was. In fact, neo-Darwinist Jerry Coyne in that science thing said still the exact same mantra, is that there's nothing weird about the Cambrian. Disparity is created by the extinction of intermediates.
And if I could just, I know you probably want to move on, but I'll just make the point that we now know from looking at developmental regulatory networks that in fact we see the same thing in the structure of regulatory networks with something very different going on in the Cambrian than we see subsequently. So there's really a structure there, both in morphology as well as in development, that's real.
Okay, we have time for one final question. Yes.
Yeah, Pat Churchland, I just wanted to ask a question about the microRNAs. And the question goes like this, that given that there are so many more microRNAs now in humans, how does that speak to this recent idea that has been suggested that Homo sapiens within the last 10,000 years has undergone a very rapid evolution?
Actually, probably nothing at all, simply because you probably transcribe somewhere in the neighborhood of 30 to 50,000 hairpin RNAs. That basically what there is just garbage. It's stochastic transcriptional noise that's thrown out into the cytoplasm, but doesn't actually regulate anything. And so it'll just be evolved away just through neutral evolution, simply because you'll lose the base pairing, which is necessary to go through the pathway. You need time. You need to be able to actually have some time for these microRNAs, the hairpins, to evolve into microRNAs by finding targets. And you raise an interesting issue, is that we don't have really any way right now of asking microRNAs that are specific to a species like Homo sapiens, because we cannot distinguish a hairpin from a microRNA unless you know it actually is regulating something important during the development of that animal. And so right now, I mean, that's a really, really gray area, because target prediction, for reasons I couldn't explain, is exceedingly difficult with microRNAs.
Okay.